After you do the PCR, you want to use gel electrophoresis in order to determine if the PCR worked. You'll be looking for one PCR band, one amplicon, for each set of primers that you ran, one and only one band in the correct size range. Tim's going to show how you prepare the PCR products for loading in the gel.
So when we're going to prep our samples for loading into the gel, we're actually going to do it on a piece of parafilm because the wax surface is really nice for mixing the loading dye and our sample together, and this way we don't have to use more like tubes or anything like that. It's just a nice, relatively inexpensive alternative, and it works really well. So what we're doing here is we're mixing. So in one of your tube racks, you have this loading dye. So what we have to do is mix some of this loading dye with some of our PCR product, and the dye is essentially a way to mark the sample and also let it kind of be weighted down in the well in the gel before we run it. That'll become a lot clearer when you're actually going to run it. So what we're going to do is put twist it down to 2.0. Alright, we're going to put a tip on there like we've done before.
2.0.
Yep, exactly. So we're going to take that loading dye, kind of, this one is, sometimes they're bluish, this one's actually got a green tint to it. This happens to be the way the brand looks. So we're going to go in and drop those 2 microliters. Yep, and then we just find a spot on the parafilm and just eject the 2 microliters there. It'll be like a very small ball. And you can just keep the same tip on. You don't need to eject it. We're going to go in and repeat that. And just find a spot some distance away, and we'll eject all of that as well. Alrighty, so that's our loading dye. You can get rid of the tip now.
And then because it's aqueous, it's hydrophobic, like it will ball up on the surface of the wax, which is really hydrophobic.
I was just looking, this one looks bigger than this one, so I don't know.
It's actually, it's okay if the volume's not perfect for this. It's kind of bucket chemistry.
Yeah, it could be a pop with a bubble in there somewhere too, making it look bigger. Alright.
And then eject it?
Yep. And then we're going to switch this to 5.0. Alright, and this time we're going to grab the small tubes, grab tube number 1, which should have your 1522R reverse primer. Alright, so we're going to grab a tip. Yeah, these are a pain to open.
Really hard to get open.
We'll go into the tube and drop the 5 microliters. Alright, and so, and then mix that in with one of the dye balls. And I do this a little bit, Caitlin. When I do it, I go in and out a little bit like this, and it kind of mixes it. Or go up and down, and it mixes it, and then I take it away. And we're going to repeat that for tube number 2, which should have the 907R primer in it.
And then just put it in there?
Yep. And just make sure you remember which one is which.
Okay.
You could always mark the parafilm if need be. ready to load the gel.
Ready for?
Before you load the gel, you have to get it ready, which means putting it into a gel rig, which is that kind of funky looking box that you see there that Caitlin has her hands over. The gel is this cloudy blob in the middle, and the blue color that you see is the comb, which is still embedded in the gel to create the wells into which the sample is loaded. So Caitlin is going to pull the comb out, and she's going to ensure that the gel contains enough buffer to run it, which means enough buffer that the entire gel is submerged.
All right, Caitlin, so you're gonna grasp the comb and just gently and firmly pull straight up. Maybe go back down again. The gel, you might even have to, sometimes you have to kind of use your fingers in a way that like, hold on.
Now, Caitlin got the gel ready and topped up with buffer, and now Tim's going to show loading the gel.
All right, I'm ready now.
Okay.
All right, so the first sample that I'm going to load is 5 microliters of ladder, which is a standard against which you can compare the PCR product that will run, and it'll give you an idea of what the size is. I'm going to load 5 microliters of this green, very green gel.
And the ladder comes, at least the ladder we buy comes pre-mixed with a loading dye, which is why you didn't have to add any loading dye or buffer to it.
And inside the gel itself, I put in a stain in there, which actually stains the DNA while it's running. So that way later on we can image it under UV light. This may be a little hard to see from where you are.
Okay.
But I'm going to, inside the wells that were created by the comb that Caitlin just pulled out, I'm going to go into one of the wells without touching the bottom or the sides, or at least trying not to. And I'm going to eject the liquid into it, but I'm only going to go down to the first stop with my pipetter. I'm not going to go all the way down because you don't want to create a bubble, which could force the liquid out. There we go. And we're going to continue to do the same with the next well, 5 microliters again, with my sample 1 and then sample 2. So on the parafilm, where we've already mixed them, I'm going to go ahead and drop 5 of the allegedly 7 microliters that I put there. And do the same thing. It will be a little hard. It is a little hard to do it at first, but you do eventually get a feel for it. And then one more time with the second sample, which should be the 907R. There we go.
So while you're loading, there are a couple things that, can go wrong. One is that the pipetter can actually go through the bottom of the gel, and you'll see it because what'll happen is when you expel, the liquid will kind of swoosh all out underneath the gel. Everybody's done this a time or two. And the other thing that can happen is if you draw your hands off the stop too quickly, the stuff that you ejected just gets sucked back up into the pipetter instead of loaded. The third thing that can happen is, you release or eject on the surface of the gel and it just kind of floats away. You can see all 3 things. It'll go underneath, it'll whoosh away up top, or it'll still be in the pipette when you pull it back out. So and you can adjust, so. All right, Caitlin's loaded her gel and now she's gonna run the gel. So she's putting the lid on the rig and it slides through these little slots just like that. Takes a little bit of force, but gentle force so you don't slosh the interior contents. And then the leads, which are red and black, going in, Caitlin? You can tell at the end. You can tell at the end if it's working. I'll tell you how in a minute. The leads go into this big box here is a power supply. Our goal here is to run electrical current through the gel. So a black lead will go into one of the black partners that Caitlin can see, but we can't see from this angle. And the red lead will go into put it close by. I don't actually know if it'll work if you spread them out. We've never tried that. I just assume you have to put them in partners. Then look and see if the thing's got, it should be set to 150 volts, which is, you can go a little higher or a little lower. It's just what we usually use in here. The higher the voltage, the faster the gel will run, the more the gel will melt because there's a bit of heat then, and the poorer resolution of the band. So if you're doing an image for a paper, you know, or you want it to be really pretty, you run it at low voltage for a really long time. If you want it to be done so that you can go home, and you know, if your experiment worked, you run it kind of faster and hotter with higher voltage.
Okay.
So that one, the run button should be the picture of the little figure running.
Okay.
And if the gel is indeed running and current is going across, there'll be some electrolysis of the chemicals in the buffer, and you'll see tiny bubbles, like a cloud of bubbles across. It's kind of pretty actually, either side of the rig. You got to check both sides to make sure that it's actually running. Because sometimes the electrical leads get kind of messed up and they don't make their connection. Look good?
Yep.
So
I see bubbles.
let's see if I can see the bubbles. If you look carefully, there are bubbles rising there, I think. I'm not sure if it's catching it in the video, but you can see it in real. Okay, and then that's it. We just note the time, and this will take about an hour to run, and then it would be ready for imaging.
So to image, and this is something that Tim did later, and he took this photo and shared it with us. And these are Caitlin's PCR products. And over up on the left, you can see the molecular weight standard, or the ladder. And then we can see 2 PCR products. And so the reason we can see them is they're glowing. And they're glowing because the DNA that is entrained in the gel has reacted with a dye called gel red. And the gel red, when we strike it with ultraviolet light, fluoresces, and we can capture it and we can see it, this pretty pink color. We got ultraviolet on there because we laid the gel on top of a UV light transmitter, and we shielded all of that in a box so that we weren't getting exposed to any UV ourselves. And this looks really good because we see 2 reactions, 2 PCR products, nice crisp bands, only 1 product, so no non-specific amplification. One of them is lower than the other. The lower one is a smaller fragment. It ran further and faster through the gel. And I know, because I know what this ladder looks like, or you know what the sizes of these ladders are, that's about where the 900 or so base pair fragment ran. And this one that's higher up is about 1500 base pairs, and it's running and traveling more smoothly or slowly through the gel. And that's about the position that I would expect it to be too. So this was a successful PCR action. And that concludes this video series.